So now we'll be moving on to the first phase of our project, which is what we call the preparation of data. And in this phase, the biology team got our samples ready to be tested and our data to be recorded, and the computational side got their database ready to actually analyze our data. And so this is just a rough timeline of the techniques and the methods that we used on the biology side of things. The first thing we did was isolated genomic DNA from a bacteria sample, followed by polymerase chain reaction, or PCR, using that genomic DNA. We then digested that PCR product using restriction enzymes and ran the product of the restriction enzyme digest on an agarose gel, and that's where we recorded our data and shipped it off to the computer science team so they could do their computational analysis. Now, the first method that we used in the biology half of this project was actually completed for us prior to our arrival at PGSS due to time constraints. And that was actually taking a sample from the food product and isolating a specific type of bacteria from that sample. And that bacterial sample is what we started out with our methods on the biology side. And the first thing we did was we isolated that bacteria sample's genomic DNA. And we did this by stripping out all of the components of the bacterial sample except that genomic DNA, and then eluting that genomic DNA into a small solution that was used in subsequent steps. Now, the first step that we did with that genomic DNA sample was agarose gel electrophoresis. Now, agarose gel electrophoresis is a process by which charged particles, such as negatively charged DNA fragments, can be separated based on their mass. And how it works is you can see the agarose gel right there. The DNA starts at the negative electrode and flows to the positive electrode based on size. So we get a nice diagram of the different sizes of products. Now, we use agarose gel electrophoresis for two purposes during this project. The first was to measure if a product was present after a reaction, and the second was to see the exact sizes of the product of whatever reaction we were testing. And just as a reference, we used 1 kilobase and 100 base pair ladders on our agarose gels. After our first round of agarose gel electrophoresis, we performed ultraviolet or UV spectroscopy. Now, ultraviolet spectroscopy is a technique that takes a very small sample of whatever we're measuring. So in this case, we use 2 microliters of our DNA sample to measure for very, very useful concentration and absorbance values that we'll use later in our calculations. Now, after our first round of UV spectroscopy, we performed perhaps the centerpiece of our preparation phase on the biology side, and that is polymerase chain reaction, or PCR. PCR is a process by which a small segment of a long DNA sequence can be isolated and then replicated thousands upon thousands of times to produce a very useful product later. Now, within the PCR reaction is first DNA polymerase, which is an enzyme that actually does the building of that new DNA, along with an abundance of nucleotides, which the DNA polymerase uses to build that new DNA, along with 2 primers, one forward and one reverse. You can see in the diagram up there. Now, these primers is what actually frames the DNA sequence that we're replicating and prompts the DNA polymerase to start at that point and then rebuild that DNA sequence to be replicated later. And those binding site sequences can be seen on the screen up there. Now, in this experiment, as Emma mentioned earlier, we are studying different samples of bacteria. And as she said, the 16S region is highly conserved between bacteria, but there are differences in hypervariable regions. So these differences can actually account for differently sized 16S regions. And as a result, since we use universal primers, these primers are pretty much ubiquitous in between the samples we used and will bind to any 16S region that we used. But because the 16S regions differed in length, or potentially differed in length, they bound to different points along that 16S region and had the potential to produce longer or shorter PCR product. So now that our biological samples have been prepared and our computational analysis tools have also been prepared, we're moving on to the execution phase of our project, which is the point at which our data will be collected and analyzed by both teams. So the biology techniques that we're using in this phase start out with restriction enzyme digest, which is perhaps the most important technique in this entire project. So restriction enzyme digests is a process by which a sequence of DNA is chopped up by these things called restriction enzymes into smaller pieces that can be used to potentially identify the bacteria. So in our experiment, we used 2 restriction enzymes. We used MSE1 on top and HPY1881 on the bottom. And what these enzymes did is they chopped up our PCR product into smaller segments, which were then run on agarose gels. And as Abby said earlier, we can use agarose gels to visualize the exact size of our product based on the size of the ladder. And we use that same process here. So what we did was we ran the product of the digest on agarose gels and measured the exact size of the product. And that was the main body of data that we shipped off to the computer science team so they could analyze it. And after we finished up with our restricted enzyme digest, we used the remainder of our PCR product to ship off for DNA sequencing. And that, as a note, was not actually intended to find an answer for the question that we were asking with this project, but was rather a way for us to definitively see if our results were correct or not from a biological perspective.